So now we're at the blackleg experiment, and this forms part of a larger project funded by BPSLC looking at various methods for controlling blackleg through cultural control. The experiment here, we have 4 different stocks of jelly with various levels of inoculum. So we have mini tubers that should be free of any blackleg. Then we have a stock which had low levels of blackleg, a stock with high levels of blackleg, and then the low stock, but then inoculated with a culture of Pectobacterium. Then on top of that, we've got 5 different irrigation regimes, so ranging from unirrigated to overwatered. And the aim of this is to understand how we can control the blackleg whilst also minimising the risk of common scab.
The 2 experiments behind me are a follow-on to the work that I was doing during my PhD. In the earlier experiments, I found that when the plant has more stems, leaf appearance occurs more slowly on each individual stem. But it wasn't clear if the slower leaf appearance was due to increased competition between stems for tuber resources or for light. So in the first experiment, tuber resources have been varied by planting tubers of different sizes. The seed was split into small, medium and large seed, and each tuber has been individually weighed out and into the egg trays so that we know exactly what resources were available to each plant at the time of planting. Then we can quantify how this affects the rate of leaf appearance. In the second experiment, plant spacing was varied to control light availability and the competition for that light. Mini tubers were planted to ensure that each plant produces a single stem so that there's no variation in the amount of tuber resource for each of those stems. Again, the plots are only just starting to emerge, so there's little to show in the field yet, but we'll be able to give you an update on Tuesday.
As in previous seasons, this is serving a really useful purpose. We actually have a crop that's grown in a very similar way each year, and that is actually giving us a benchmark against which seasons can be compared, and also the other crops in the field. So that's one of its sort of key functions. Another sort of really important function is to test ideas, hypotheses. So one of which I've mentioned before is this effect of soil texture on soil emergence. And we think this may be due to something to do with soil temperature. So again, this year we're looking at soil temperature in the plots of different soil texture. Another sort of key role of this, of the reference crop, and it's something we've been thinking about for many years, is perhaps to have a better understanding about why we have yield variation from season to season, and also different responses to nitrogen from season to season. And what we have here this season is perhaps quite detailed examination of soil properties at the time of planting and the time of crop establishment. So things like the VES assessment, we've taken some bulk density cores, we've taken some cores for water soil aggregates, we've also got penetrometer resistance data. So hopefully after a few seasons, we'll get a good idea about what kind of soil physical characteristics may have an influence on crop yield potential and also response to nitrogen. Another sort of couple of roles this experiment's serving is the russet Burbank crop plots in the reference crop. They're sort of being run in parallel with some similar plots in Washington State with Mark Pavak. And these two sort of series of plots in the UK and in Washington State helps us better understand the physiology of the potato crop, but also gives us a better understanding of what environmental factors control yield potential. So hopefully we'll get some really interesting data off that this year. And the other thing we're using this year for the reference crop is we're just piggybacking a small pilot study we're doing with McCain, looking at sustainability or factors controlling sustainability in crop production. Another benefit of the reference crop, which, as I said before, is a crop that's grown in a similar way year in, year out, is the opportunity to test new technology. So one of the factors we've mentioned before is using electrical conductivity scans. And as I've mentioned, we're using those electrical conductivity scans to better understand the variability of soil texture we see within a field and also within experiments. And for example, in this experiment, the 2021 reference crop, we've altered the block structure to take better account of the variability we see in the EC scans. The second bit of new technology we're actually working on at the moment, which you can see being installed in the background, are some in situ nitrate sensors, which will remotely upload their data onto the cloud. So these are being installed by CUPGRA, and hopefully if all goes to plan, we'll actually see time courses of nitrogen fluxes within the soil profile. And I think this is going to be a real step forward in actually bettering our understanding of nitrogen and nitrogen dynamics in potato crops.
The AHDB Agronomy and Storage project is now in its final year, and in light of the limited variation that we found between stocks stored commercially and at Sutton Bridge, we're focusing on a different aspect of storage this year. This year we're focusing on temporary seed storage just before planting, which occurs on farm, and we have stored seed under good conditions, so in a well-ventilated crate at 3.5 degrees, and also poor conditions in a jumbo bag and ambient temperatures. Then we have planted the seed both here at CUPGRA, at Spot North in North Lincolnshire and at Spot West in Herefordshire, to see what effect the differences in site can play into the variation in crop performance. Additional treatments have been imposed at CUF, so we have not warmed some of the seed, we have allowed some of the seed to get wet, and also chilled some of the seed to explore further aspects of poor storage practice and how that affects subsequent crop growth. As you can see, the crop has only just emerged, but already there are some differences between the different treatments, and I'll go into more detail on these at the Members' Day itself, when we know precisely what emergence has happened.
Hello, my name is Gerard Bishop. I provide an independent assessment of new varieties at CUPGRA. And this year I have 4 experiments in the main field, F37, and that is to help provide independent assessment of some new crisping varieties for PepsiCo and the third-party breeders. And this includes analysis of disease, susceptibility, providing an assessment of the nitrogen requirement, and also providing an assessment of seed rates that are required for those varieties to help maximise their performance in the future. Where we are here now, this is in field F43, and this is where we're carrying out an independent assessment of Hermes Plus lines. Hermes is obviously a crisping variety, and these have been engineered to have blight resistance, to have less sugar accumulation during cold storage, and also less discoloration due to bruising when bruise occurs. So this work is being carried out for Jonathan Jones's group up in the Sainsbury laboratory, and is part of funding from the BBSRC and UKRI. And this is a continuation from last year's successful experiment with Maris Piper lines. As you can see, this is the trials being carried out in a fenced area. So this is part of a DEFRA release, which is a consent from DEFRA, and so we need to be in a restricted area, and so hence we're in a fenced area, and it's also away from the main field because we have to be away to prevent any pollen going between the 2 sites. This year we'll be assessing 9 different transgenic lines and all in comparison to 3 controls. 3 controls are Hermes, which are just a different seed size. And we're trying to provide an independent assessment of the agronomic performance of these lines. They might be different due to the way that the integrated DNA or in the production during tissue culture may have changed some of the made some somoclonal variation within these lines. This year there's not much to be seen at the moment because emergence is only just starting to take place. It was planted on the 28th of April and today is the 3rd of June, and so hopefully by the time when we come to the CUPGRA meeting and online on the 15th of June, I'll be able to provide you with more of an update and some slides of showing where the crop is and kind of with emergence and everything else. And so I look forward to seeing you then.